Hi, my name is Alex Serrano, and I'm here from the University of Colorado Boulder, and we use nanopore technology to sequence a big pool of novel bacteriophage genomes and performed some analysis on their sequences. So I guess to start off, I'm going to give a brief introduction of nanopore. It's a type of long-read sequencing where we have these adapter ligated genomes and they pass through an array of these little pores on a big flow cell. And passing through these pores causes a change in an electrical signal, and this electrical signal can eventually be converted into a genome. And the biggest reason we use nanopore is the fact that it's actually cheaper and it's a lot quicker to use than sending it all the way to Pittsburgh for Illumina sequencing. And we wanted to see if, potentially this could be something that could be used here at the University of Colorado to assist in the understanding of the C-phage bacteriophages. And just a brief introduction of a brief overview of how the whole process works. I'm going to go over here to figure 1. First part here is we have the library prep loading. We gave unique barcodes to all the different phage samples, pooled them all together, ran them through the sequencer, and that gave us the sequencing that spits out unique electrical signals. We performed a base calling, which turns that electrical signal into unique raw reads, then split them apart, sorted them all by their barcodes, and got these FASTQ files, and these are raw FASTQ files. And from there, they went to an assembler, and the assembler performed a correction step. It used de novo read graphs to assemble all of them into our consensus sequences, did a little polishing, made sure they looked nice, and then produced these FASTA consensus files. And then from there, I'll turn you over to my colleague Charlotte, who did a lot of the post-assembly analysis work.
Hi, yes, thank you, Alex. So out of all the sequences that were run through this method, 4 in particular were B1s, which I decided to investigate further. By B1, I mean a part of the B1 subcluster of bacteriophage. So the first thing I did was took each of the samples sequences and stuck them through a full genome blast on NCBI. And this, of course, resulted in a list of top results for each of the sequences. And I took the top result for each sequence and then stuck it into a dot plot. So you look down here at figure 3A, you can see you have my samples, which were lovingly named Fuzzy Squirrel, Fuzzy Watermelon, Lava Girl, and Muddyfoot, and stuck them against their top blast results here in black. So looking at this figure, you can see that it's very fragmented. You see particularly here between Lava Girl and Muddyfoot, you've got this giant leap, and other points there is sort of weird overlaps that just don't make sense for sequences that should be in the same subcluster. So after conversing with my colleagues, we realized this might be something to do with the start site. So I went back to just those fully annotated genomes that I got from NCBI, from the BLAST search, and stuck them in Famarator. Now you can see up here in figure 2A, that at the start of their genome, they each have exactly the same sort of combination of genes, which is a small forward gene, a long forward gene, 3 small forwards, and a reverse gene. Now I thought, okay, this is a pattern which I can use to try and identify the start, or the official start, should it be, in all the sequences we got from Nanopore. So looking down here at figure 2B, this is Fuzzy Watermelon in particular. And I scrolled along the genome in DNA Master until I found this pattern. So you can see the small forward, the long forward, the 3 small forwards, and then the reverse. So I thought, bingo, this is the correct start site. So I then took that, which in Fuzzy Watermelon particularly falls at gene 28. I found exactly what nucleotide it was by using DNA Master to look at gene 28, then went to Benchling, put the whole sequence in, and Benchling allowed me to effectively do a copy and paste where I moved the start to the start and the end to the end, depending on the different sequences, as each of them found the actual start site to be a different location. So once I did that, I then went back to making dot plots once more. And as you can see, figure 3B here, this is a perfect looking dot plot. There's no more fragmentation in the samples. And in fact, if I took away the labels for which are the samples and which are the fully annotated genomes from Illumina, you probably couldn't tell the difference anymore. So from this, we can conclude that our results suggest that nanopore sequencing is a method that can produce sequences of sequences with accuracy of to a similar caliber of Illumina sequencing. And with that, I'll send you back to Alex to explain the rest of the details.
Thank you, Charlotte. And just to kind of bring this all back, nanopore sequencing offers an inexpensive and effective method to quickly produce these high accuracy sequences for educational purposes. And so for some of the future directions, nanopore does have some drawbacks. Probably one of the most notable ones is that it does have issues with accuracy in repetitive regions. And we want to examine the effectiveness of next generation nanopore chemistry kits and nanopore flow cells and see if they can improve some of these known problems. And furthermore, we want to use nanopore to sequence additional phages that have Illumina reference sequences so we can build essentially a better profile of their accuracy for nanopore compared to Illumina. And with that, I'd like to give special thanks to Dr. Giles, Dr. Filman, and Dr. Chong for all of their help with this project. And I'd like to thank you all for your time.